If you have a problem, if no one else can help, and if you can find them, maybe you can hire the A-Team.
What goes up must also come down. So there's a saying. The same goes for nutrients. The nutrients that drop from the trees each year must find their way back into the tree. But the trees cannot readily absorb their own dead leaves. This is where decomposers come in. They are organisms that break down the dead leaves into by the plant absorbable elements. Now, the ecological importance of this process is that it ensures that nutrients are returned back to the soil, from which they can be reabsorbed, forming a critical link in the nutrient cycle. Decomposers also create a suitable habitat for many other organisms. Forest soils also hold a lot of carbon. Knowing how these systems function might help us increase the storage potential of the leaf litter. For now, our focus is on millipedes, as one of the most common decomposers. As a result of their large ecological role in nutrient flux, millipedes are considered ecosystem engineers. This makes it important for us to understand their ecology, so we can conserve millipedes the best we can. Millipede abundance and diversity are influenced by many factors. One important component is the tree assemblage. The diversity of trees is likely to positively influence the millipede diversity, since more niches are created when more different types of leaves are present on the forest floor. The different leaves all differ in characteristics, including organic carbon, nitrogen, and phenol content. Also, the litter layers differ in fragment size and leaf litter thickness. Carbon and nitrogen are important since a higher carbon-nitrogen ratio slows down decomposition. Higher nitrogen contents increase the nutritional quality of the leaves. Higher carbon content often indicates a high amount of molecules that are difficult to digest. Studies also showed a high preference of millipedes for litter with a high nitrogen content and a low carbon-nitrogen ratio. High phenol content also negatively influences digestibility, although it does not always explain preference. Although one of the first decomposers, millipedes do prefer older leaves. This could be correlated with the fragment size of the leaves. Litter thickness can harbor a more diverse habitat with more layers, also influencing the microclimate, including humidity. A thicker leaf layer would hold more moisture, and the temperature would be more stable and cooler. Predators have a negative effect on millipede abundance, and presence of predatory insects is also influenced by the microclimate.
The fieldwork study was carried out in Oostereng Forest. The Oostereng Forest Estate is located in between Renkum and Bennekom and is owned by Staatsbosbeheer. The Oostereng contains a large variety of forest stands. Having many different types of forest in the same area limits the random variation and increases the chance of detecting differences between various types of litter. The fieldwork setup included 24 different plots. The plots were selected to contain various forest types, including beech, oak, Douglas, pine, and larch. Our focus is on explaining the millipede abundance and diversity. The independent variables that were measured are tree species diversity, depth of the litter layer, fragment size, moisture content, and nitrogen and phosphorus content of the litter. We picked a random point, a starting point of the transect line, and a random number for its direction. From this point, a pitfall transect line was made. A gap of 2 meters between each pitfall was used to eliminate distance-dependent catch change. The pitfalls were made from plastic cups that were dug in the ground. The pitfalls were placed so that the top of the cup was underneath the litter layer, to make the millipedes in the litter layer fall in. We filled the pitfalls with a mixture of water and methanol. This prevented the millipedes from crawling out of the cup and being predated by other trapped animals. A total of 480 pitfalls were placed over the course of the experiment. The pitfalls were left for 24 hours, after which they were emptied and the number of species of millipedes was noted. Among the other things measured was the tree species diversity. This diversity was determined by measuring the amount of trees that overhang within 1 meter of the transect line. This measurement represents the tree diversity. The diversity of tree species per plot and the total amount of millipedes per transect were calculated using the Simpson's Biodiversity Index. The depth of the litter layer was measured from the top of the litter layer to the sand layer. The litter samples were collected at 3 locations along the transect line. These samples were analyzed at the lab. First, the samples were put in the dry oven and the dry weight was measured. After this, we were able to measure the fraction size, which was measured using a 4-layer sieve. After that, the percentage of each particle size group was measured. A chemical analysis was carried out to measure the nitrogen and phosphorus content of the leaves. Over 500 millipedes were caught in the field, out of which 9 were identified. Polydesmus angustus was the most abundant with 320 individuals. According to literature, the species is very common in the Netherlands, especially in the east and the southeast of the country. It mainly occurs in broadleaf and coniferous forests. Furthermore, we found 100 individuals of other Polydesmus species. To explore the relationship of different environmental variables on the millipede species, we used the program Ecological and performed a principal component analysis, a PCA. The first axis, with the eigenvalue 0.45, is mainly correlated with the amount of litter of 1 mm fragment size. Other environmental variables, litter depth, humidity, other litter fragments, and nitrogen content are also related to axis 1. 3 of the millipede species have a strong positive correlation with this axis. Several other species are negatively correlated with this axis. This is as predicted, since different species occupy different niches. Ecologically, this is important because it permits the existence of different species on limited resources within a given range of the ecosystem community. The second axis, with the eigenvalue of 0.19, is mostly correlated with the tree species diversity. Most of the species are positively correlated with tree species diversity, which is according to our hypothesis. The total explained variation of these two axes is 64%. After carrying out the PCA, we carried out a regression analysis to test for a significant relation between millipede abundance and diversity. After carrying out the PCA, we carried out a regression analysis to test for a significant relation between millipede abundance, diversity, and the different environmental variables. To achieve a normal distribution, we did a natural log transformation. A backward regression was carried out, after which millipede species abundance showed a significant relationship with tree species diversity and litter fragment size of 1 mm. These 1 mm fragments are commonly found in old leaf litter, which has been broken down over time. This implies that millipedes prefer older leaf litter. Smaller fragments also provide a larger surface area, which is easier to utilize by decomposers. More millipedes occur in areas where there is a higher diversity of tree species. This can be explained by different leaf litters, which provide different nutrients in appropriate balances for millipedes. It also provides a variety of living conditions for other litter fauna on which millipedes depend for various ecosystem services. So, tree species diversity also ensures a healthy functioning leaf litter ecosystem. The same applied for the millipede diversity. At first, no significant relation was found. However, after a forward selection, a significant relation was found between the fraction of 1 mm leaf litter fragments and the millipede diversity. Ecologically, this makes sense because smaller fragments provide a favorable habitat and a higher availability of nutrients. This is because millipedes depend on other organisms for many biological processes, such as digestion in millipedes' digestive tract. This is mainly done by microbes whose biomass is increased by smaller fragmentation of leaf litter. Due to an outlier in the scatterplot, the tree diversity and the millipede abundance was significant. However, when we removed the outlier, this relation was not found. Therefore, we assumed this was a false significance and thus rejected. The millipede diversity and the 1 mm fragment size do not show a very clear linear regression. This might be the case because normally a higher percentage of 1 mm leaf litter will positively influence millipede diversity. However, at a certain point, other environmental variables such as humidity and litter depth will limit the millipede species diversity. When these were removed, the graphs show a better linearity between millipede species diversity and 1 mm leaf litter fragment size, and the regression line would have a better fit. This shows that although the variation is high, there is a positive relationship. From the PCA analysis, it appeared that the tree species Polydesmus denticulatus, Polydesmus augustus, and Brachydesmus superis were strongly related to the amount of litter with a 1 mm fragment size. Regression between these species and the 1 mm fragment size resulted in the following graph. From these species, only Polydesmus denticulatus has a significant relation with the amount of litter with a 1 mm fragment size.
The goal of the lab experiment was to determine the preference of two millipede species, the pale millipede Glomeris marginata and the flatback millipede from the genus Polydesmus. In the lab, the effect of confounding factors can be reduced. The preference of these two species was tested with the litter of three tree species: European beech, English oak, and Douglas fir. Each millipede was given a choice for two different litters. This totals up to three different choice combinations from which the millipedes can choose. Two different species of millipedes with three different combinations resulted in 6 different combinations. To check for an interaction effect, the millipedes were also put together to see whether the presence of another species might affect that choice. These 9 different combinations were each copied 27 times. The leaf litter was dried and then equally moisturized, after which a little was put on one half of each petri dish. The complementing litter was then put on the other half, making sure the amount of litter on each half was equal. After all the petri dishes were filled, the millipedes were randomly assigned to each of the treatments. Following this, each petri dish was randomly put in a box. The box was put in a dark place and left overnight. The next day, the position of each millipede was noted. To detect a preference of litter type, a binomial test was used with the expected proportion being 50%. First, we tested the single species separately per treatment. In all the combinations of millipede species and litter treatment, no significant difference could be found. After this, the same test was done with the double species experiment. Except for the choice of pale millipede between oak and fir, there were no significant differences. Since 12 tests were done after each other, chance could generate significant differences in one of the tests. Therefore, a Bonferroni correction was done, which rejects the null hypothesis with a p-value lower than 0.004 instead of 0.05. With this correction, none of the tests showed a significant difference. Here, one of the experimental results is shown in a graph. As said, it is not significant, and in this case, the flat millipedes chose oak a bit more often than beech in the single species experiment. The other single species experiments show a similar result, without a true preference. For the double species experiment, one treatment showed a significant result before using the Bonferroni correction. In this experiment, pale millipedes that were together with flat millipedes seemed to prefer oak over Douglas fir. The other double experiments showed similar results as the single species treatment. For the pale millipedes, the preference showed the same pattern in the single and double experiment, where it seemed to prefer oak over fir, but beech over oak. So no preference for leaf species could be found in the lab experiment, while we did expect a difference. In India, the food preference of pale millipedes was correlated with nitrogen and carbon content. The study showed millipedes prefer leaves with a high nitrogen content and a low carbon-nitrogen ratio. Although this is not the only component influencing this. Also, low phenolic content, high calcium and phosphorus content were preferred. Phenols are often hard to digest, although some species can process them more easily. It is also interesting that food conversion efficiency does not always reflect the preference of millipedes. This is likely due to a lot of factors influencing this choice. The leaf species we chose in our lab experiment also differ in chemical content. In general, oak leaves have the lowest carbon-nitrogen ratio, followed by beech. Douglas fir has the highest. But differences between sites and time of year occur. Beech leaves also have more phenols than oak leaves. In our analysis, we found small differences in nitrogen content, but Douglas has a lower phosphorus content. According to these characteristics, oak seems the most suitable for millipedes, followed by beech, with the last being Douglas fir. So we expected most millipedes to choose for the oak leaves, but we did not find any preference. Although not significant, the pale millipedes did choose oak over Douglas. The flat millipedes did not show any indication of preference. Polydesmus angustus is known to be a generalist, which does not show a high preference for its outer layer. Furthermore, although tried, not all other variables, such as moist and fragment size, could be equalized between the leaf species. Maybe they did not choose for the food, but for a nice spot to sit. Since no real preference was found for a certain leaf species, also no competition occurred. In the wild, millipedes share an abundant litter layer with multiple detritivores. If they have their own niche within the litter layer, changes of competition become smaller. In the lab, often the two species were found in the same litter type, so there was no indication they avoided each other. As mentioned, the preference for nitrogen and phosphorus-rich litter could influence the millipede occurrence. But in the field, we did not find any correlation between these chemical components and the millipede abundance nor diversity. The relations of millipede abundance and environmental factors can be present, but it depends highly on location. In one research, they did not find any relation with 12 different environmental factors in a forest, but did find correlations in a plantation, such as humidity and carbon content. Nitrogen content of litter is said to be the main ecological factor controlling the distribution of millipede populations at local level. Not finding this correlation in our field study can be caused by the generalist species that occur throughout the whole forest or other environmental variables that were not measured. such as specified microclimate variables. One microclimate factor that we did measure was moisture content of the leaves, but this also showed no correlation. This correlation could also be species dependent, since moisture content plays no part in the habitat preference of Cineroides punctatus and Glomerus marginata. But it does influence Julius scandinavius abundance. No correlation between millipede abundance and diversity with the chemical components was found in the field. This can explain why the millipedes in the lab showed no leaf litter species preference. However, in our research, only tree diversity and leaf litter fragment size do play a role in the diversity and abundance of millipedes. So how does this correspond with our research questions? To come back at the research question, what is the difference in diversity and abundance of millipedes between various types of leaf litter? Tree diversity as well as leaf litter fragment size both increase the millipede diversity and abundance.
I am a millipede, I am amazing. I command you to gaze upon my face. You'll never find someone charming as I am. I'm the swankiest bug out in space.